Welcome to Biology Made Easy. In today's lesson, we are explaining the chromosome numbers during the cell cycle. Welcome. The chromosome number at G1 is 2N diploid. The chromosome number at G2 is 4N tetraploid. The chromosome number at the beginning of mitosis is 4N. At the end of mitosis is 2N. We want to explain that. Well, these are two newly formed cells. After cell division, they are 2N in diploid organisms like human beings. Now, these two diploid cells enter G1 for growth. This cell differentiates and becomes a specialized cell, like a muscle cell. This cell is committed to divide, and so it enters into S, the state where DNA replication occurs. Now, at interface G1, let us use this as an example. This is a cell 2N. It means it has two copies of each chromosome, 2N. Well, one of the chromosome is maternal. I labeled it A. The other one is paternal, B. What it means is that in a human being, we have 46 chromosomes. It means we have 23 pairs. So if this is a cell, I have labeled 1, 2, 3 pairs. So what it means is that this chromosome, maternal, has a paternal one. This maternal, paternal, maternal, paternal. And so it means that these two, the genes on the maternal, are the same genes on paternal. If the gene for maternal is T, the gene for paternal will be T, P, P. That is why there are two copies of the same chromosome. And this is what gives us the 2N diploid. Two copies of the same chromosome having the same genes or similar genes. So we have two copies, DN, at G1. So I have these two chromosomes, A and B. They are homologous. I have drawn it as this DNA, and you know that the DNA, when it comes into the S phase, the DNA is going to replicate. So the DNA for A replicated with the centromere attached. So you have one sister chromatid. The DNA B also replicated, and so you have one sister chromatid attached at the centromere. So in the S phase, after the duplication or replication of DNA, you actually have 4 chromosomes. 1, 2, 3, 4. So you have 4 copies of each chromosome. One here, one here, one here. So these 2 chromosomes finally becomes 4. You see? 4 copies of each chromosome, because the 2 chromosomes, they are actually one type of DNA because it carries similar genes. So by the time it replicates, it's no more a diploid 2N, it's a tetraploid 4N. So that is what is happening after the S phase. So by the time the cell enters into the G2 phase, the cell is a tetraploid. So it enters into the M phase, mitosis stage, being a tetraploid like this. During mitosis, the centromere holding the 2 sister chromatids break. So it will break here, and this one will become one chromosome, one chromosome, one chromosome. This one too will break one chromosome, one chromosome during mitosis. And by the end of mitosis, there are 2 nuclei. Each of them is a diploid because this A, the A will break, centromere will break. One A will go into one nucleus, another A will go into another nucleus. The B2 will break at the centromere. One B will come here into one nucleus, another one will come to another. So you end up by having the diploid nuclei. These diploid nuclei, mitosis is chiriokinesis. The phase that follows is Cytokinesis, where the cytoplasm divides. So here the cytoplasm has divided and you have two cells, each of them with two chromosomes, and it's 2n because it's two copies. Well, this is where we call it a day. If the lessons have been useful, please give us a thumb and kindly subscribe. Thank